the conference room, and also thank you everybody who is connected from another place. Today we have a new session of the Friday Talk. The speaker today is Carmen García-Comas. Carmen did the study in biology in Oviedo in 2004, and then she did the PhD, which was presented in 2010 in Villefranche in Naples, at the Station Zoologique in Naples. And then she has been as a postdoc in the Far East for many years until she came back here to Catalonia in 2018, and in 2019 started here at the ICM. She summarized her experience on an interest in understanding the community structuring, diversity maintenance, and the functioning of aquatic systems. And she uses for that large data sets and applies theoretical ecological theory. So thanks for accepting to give this talk.
Thank you. Okay, I will take off the mask. So yeah, as Ramon said, today I will present our last work with Pedro Cermeño, who is the PI of Inditec project, an Explorer project from the Plan Nacional. And this should be a presentation in 4 hands, because the paper was written in 4 hands. But Pedro is flying now, so he cannot be here. And then actually this work has been done in many hands. You will see it's a big collaboration, so this was very nice to participate in this kind of real collaboration between experts of very different fields. So in Inditec, we have modeled the regional diversification of marine benthonic invertebrates in the last 541 million years. And before presenting the work, I will present myself briefly, because I've been hiding in the corridors of ICM as much as I could until now. So my career in research started with an internship, like many. Some of you at ICM start like this also. So I started an internship right after my degree in biology in Oviedo. And this internship was first in the Instituto Español de Oceanografía in Gijón, and then I moved to the Plymouth Marine Laboratory in England. And I was doing internship things, so sampling and sample processing and these kind of things for plankton. Then I got a fellowship in the Eurasians Marine European Pool, and I went to do a PhD between the Laboratory of Villefranche in France and the Station Zoologica of Naples in Italy. And in this PhD, I focused on long-term time series in the Mediterranean Sea, and we analyzed big groups of mesozooplankton. And I focused especially on the size structure of copepods along 30 years in the Mediterranean. So we were basically, among other things, checking if size diversity was a good indicator for changes related to environmental forcing. After finishing the PhD, it was the beginning of the European crisis, so not much money. And I got offered a very short contract as a technician for a private company that was actually the one manufacturing the machine that I used in my PhD. So during my PhD, we developed, I was in the process of developing this machine that is just a scanner of zooplankton, water samples, just to measure the size of individuals very fast. And so after the PhD, I went for a couple of months around the Mediterranean laboratories, setting the machine and teaching the people how to use it. This lasted just 2 months, but I already had an offer to go for a postdoc in Taiwan. So it's not China, but near China. And so at the beginning, I was a bit scared of going so far away from Europe, but I just said, okay, just for 1 year. And it took me 3 years of postdoc. It was really, really interesting work, because I landed in a group led by Qihao Xie. He's called Zach in the American field, and he's super good in theoretical ecology. and ecoinformatics. So I learned a lot in that period. And there we analyzed, I focused on analyzing also the size structure of plankton, but this time from nano microplankton to mesozooplankton. And we tried to understand if the size structure of nano microplankton and mesozooplankton separately had an effect on the trophic transfer from these two groups. All this was work from observations in the field, in this case in the East China Sea. And then I wanted to come back to Europe and start competing with the Europeans. But my boss there, so Zach, said that there was a postdoc position in a very good group, again, with a big project on modeling this time. So they were inviting interviewees to Tokyo, and I thought, oh, this is a nice opportunity to visit Tokyo. They pay the trip, so I just go. But the surprise was that I got selected for the position, and then it's very difficult to say no. So I said, okay, just for 1 year, and then I will come back. And it took me 5 years to come back, because it was really nice group, and it was modeling, which I wasn't familiarized with. So again, I learned a lot. But there was a problem that I was a data analyst, so I didn't have many papers in collaboration because they were modelers. So I'm in many papers as acknowledgements, like ideas, but I didn't model at that time. Anyway, I enjoyed a lot Japan, and I was in Jamstec. It's a very good lab with an institute with a lot of money, resources. And the group I joined was modeling plankton size structures, so the theme, the ecological theory behind I knew about. And it was an optimality-based model. And so I was feeding the model with data collected in the sea, and then with ideas from theoretical ecology. And then on the side, they gave me a big data set, the data collection of zooplankton. This model was on microplankton, on phytoplankton, but okay. And I could play with this data. So in this postdoc, I decided to focus on translating this historical data on species counts into species-level functional traits, like if the species is small or it carries their eggs or different traits, and try to see if the species-level functional traits had an effect on the species abundance rank, had an effect on which species would dominate in a community or not. After this, finally, I came back. I came back to the University of Vic. There was a woman doing things on size structure, and she was reviewing one of our papers, and well, she was interested in collaborating, and at the end, I came back. And I was in the University of Vic teaching as research assistant, and since then, I collaborate with them on analyzing, again, the size structure, but this time in fresh water, from microinvertebrates to fish. And especially we are working on the role of size structure on recreational fisheries management. And finally, in 2009, I arrived to the ICM. I will tell you what I do at ICM. But before, I just want to give you a small snapshot of this work in Japan. Now it's under review in Global Ecology and Biogeography, and if you type this title, you can find it also in bioarchive, an older version of the paper. Anyway, this is just a snapshot to say that maybe I can do a tema in the future about this, because even if it's on copepods, it's on species community structuring, and maybe it's interesting for your microbial data. I just developed a very simple method on analyzing this effect on functional traits on the abundance distribution of species, which species dominate and which are rare, depending on how they are, which traits they have. Anyway, let's go to the point of this presentation. So I arrived at ICM in 2019. Pedro Cermeño got me in this Inditech project, a very exciting project for me. So I was a postdoc at Inditech. This is an Explora project. And we have developed the first regional diversification model of marine invertebrates along the Phanerozoic, from the time for evolution and the speed of evolution, depending on the environmental conditions in the ocean. And since March of last year, I've been project manager in Prodigio. It's another project from Pedro, that is an H2020 project, where we are sampling microbial communities in algal ponds and in anaerobic reactors to analyze the nonlinear dynamics of interactions, microbial interactions in these communities, and try to understand with multi-omics and chemical fingerprinting and other measures, try to understand why these communities tend to crash in their production, so biomass production of algal and digestion from bacteria, in order to try to predict these crashes before they occur. And the ultimate goal of Prodigio would be that we try to make more sustainable the production of biofuel. But anyway, the focus of my presentation today is the Inditech results. So why we modeled this diversity in the last 500 million years? Well, because we know about the diversity has been increasing since 500 million years, and we know more or less how it has increased from this, on the left, global diversity curves from fossil records. And from these fossil records, there's been a long-standing question about how this diversity accumulates in our planet. There's a school of researchers that says that the diversity has accumulated along these 500 million years in a logistic way, in an equilibrium model. That's it, diversity accumulates until K, carrying capacity, so until the diversity that the Earth can maintain. And there's another school of researchers that says that diversity actually in the Earth has accumulated in a non-equilibrium model, so in an exponential way. That is, that we can ignore, the limit is so far, the limit of the Earth, or the carrying capacity, that we can ignore it, and diversity is just accumulated exponentially. And these two schools base all their ideas on these fossil records, that they are global curves, different global curves from different ways of putting together all these paleobiologist counts. And these two schools, they base their points, for example, the equilibrium model say that the plateau in the Phanerozoic is a very good indicator of this carrying capacity of the Earth. And then the others say the opposite, that since the end of the Cambrian, the diversity has increased exponentially, and so there's no limit. Okay, but diversity varies dramatically among geographic regions, and actually each region has its geological and environmental history. So it's difficult to address this big question from this global fossil curve. And also, fossil data are patchy, so are kind of spatiotemporally biased. Here I show you a plot from Benson et al. In 2021, where they plot the top figure is about the marine ocean. They plot the position of the shallow coastal area where diversity accumulates in the ocean, and on top they plot the red dots as the fossil records found. And then the line is just the trend of these fossil records. And you can see that fossils are mostly from North America and Europe. So we have a biased view of the past diversity in the ocean. And then I told you that regions have different diversity, but they also have different history. This is a simulation of plate tectonics from Pangaea from 250 million years ago from our collaborators from Earthbite Group. And the color represents the age of the seafloor, and the seafloor is constantly renewing. From here, from the mid-reach of the oceans, it renews here, and then it goes spreading the oceans until they subduct and disappears this ocean crust below the continental crust. So the Earth is very dynamic, and so the diversity has, if we want to model diversity in the past, we need to take into account all these dynamics, all these plate tectonics. Because of the continuous renovation of the seafloor, the maximum age of the floor rarely exceeds the 250 million years of age. Then it disappears, it subducts. And the seafloor age is spatially heterogeneous in the global ocean. So here you can see the violet, dark violet is the place where new ocean crust is creating, and then the yellowish is the oldest ocean floor, seafloor. Also, the sea level, so the paleoelevation, determines which parts of the continental crust is underwater. So we can also determine how long the coasts have been flooded underwater, so they can keep diversity. Both the sea floor age and the time that the continental coastline has been underwater give a different time for diversification in that floor at a given time. So what we see today is the integration of all this time. And then every region in the global ocean has different environmental conditions. So these are data from our collaborators from Sea Genie model and Earth System model for paleoclimate, where we can know, very simplified, how the environment was spatially distributed in the past until today. And in the left is the food that we will consider in our model, that is just a particular organic carbon flux to the bottom of the sea, arriving to the bottom, and then the temperature in the bottom of the sea. So these different environmental conditions give different pace for diversification. If you have enough food and high temperature, you will have faster diversification, so faster accumulation of diversity. So to catch up the main point, there are big questions arising from fossil records, but fossil records are patchy spatio-temporally. So we have decided to better understand how diversity accumulated in the ocean in the last 540 million years. We decided to build the first regional diversification model by coupling a paleogeographic model, G-plates, that gives the time for diversification in the different areas of the ocean, and an Earth System model, Sea Genie, that gives the pace, the speed of diversification in the ocean. So we built a very simple diversification model in which diversity accumulates regionally at a pace, the net diversification rate, rho, given by the interaction effect of food and temperature. Diversity with temperature increases in an exponential way, following an Epley curve, and diversity increases with food in a Michaelis-Menten way. The interacting effect of both gives the speed of diversification, so the speed of accumulation of diversity. And we tested like 40 combinations of parameters, and at the end we got the best combination by comparing our model output with the fossil global curves you will see later. And then we gave a range of diversification rates from a bibliography, from a paper. It's from Stanley in 2007. He checked how many genera appeared in each X million years in the fossil record. So from there we get this range that I put here, but it's not important for this. It's just detail of the model. And then in order to answer to the question of the Earth is near equilibrium, so it's in a saturated logistic way, or is far from equilibrium, and we can simplify to an exponential way of diversification, we model the two ways of diversification. In the logistic model, we have to take into account the carrying capacity of the environment, and so diversity increases first exponentially with the interaction of food and temperature, but then when it's approaching its carrying capacity, if diversity limit that the system can contain, it slows down until it stops on the top near the KF. KF is the effective carrying capacity, and this is given just by the food available in a certain region in the ocean. Vale? To test for this hypothesis, we set the range of this carrying capacity very low, between 4 and 16 genera. So we approach this saturation and see how the global curve will be with this hypothesizing that there's saturation in the Earth. And then we do in parallel the exponential model, which is just a simplification of this. We just assume that the carrying capacity is infinite, and then the equation stays like this. Diversity increases depending on the interaction effect of food and temperature. And then, so imagine these curves in every patch, in every area of the ocean at every time will be with a different slope depending on the food and temperature. The more the food and temperature, the steeper, the fastest the way of diversification. Okay, so as I said, we have a paleogeographic model, the G-plates, that gives the time for diversification. It tells you the age of the ocean at every 5 to 10 million years. And it gives it in points. They just seed the ocean with points, and then they track their position as the plates move and as the ocean opens up, is regenerating from the mid-ridges. So it's tracking like 44,000 ocean points at every time frame for 82 time frames. And then we are also tracking 6,000 coastal points. These coastal points are more static, so the continents move and break, but they don't disappear like the ocean crust. And we follow also these points to see if they are underwater or below water. So our model starts at 541 million years ago, imposing to all these points at the beginning a diversity of one genus. And then as these points move around and they get different environmental conditions, diversity accumulates at different pace. In the case of the ocean, every time a new crust is forming in the middle of the oceans, we give this new point a diversity equal to 1. And then when it disappears, the crust, so these points disappear below the continental margins, we make it disappear, the diversity carried by these particles. And then in the case of the coastal points, at the beginning they start with one genus, and then they start to accumulate. And when the sea level drops, this diversity disappears. But when the sea level goes back up, diversity doesn't start at one genus, it will start at the diversity of the closest coastal point. This is just to assume recolonization in coastal environments, okay? And as I said, to these points, we give a pace of diversification depending on the environmental conditions from this model, the CGNI model. So this model is an Earth system model. So it's based in a 3-dimension ocean circulation model coupled with a 2-dimension atmospheric component and sea ice module. And the model has 17 layers in different depths until the depth of 6,000 meters. And they have well, I wrote some specifications, but maybe I don't need it. We have a compartment of plankton, so we can account for the remineralization of nutrients and everything. And at the end, what we want, the output that we need is the temperature at the bottom and the POC that arrives to the bottom. And because this model is very computationally, it's very, very heavy, they gave us 30 time frames with this spatial resolution, 36 by 36 grids. And every frame is disconnected from the others. They just model this kind of configuration of continents and ocean, and they run it for many years, like 6,000 years, and at the end, they obtain the last year as the output for our model. So it allows for the deep ocean to get the equilibrium and all that. So it's quite complex, this part, and it's made by these people. The CGNI modelers. So we just interpolate into our points the food and temperature from these maps. So we get the pace of diversification. And finally, we have to impose from fossil diversity curves the big extinctions. Why? Because the mass extinctions have many interacting mechanisms that are very complex. Even we don't know some of the extinctions, we don't understand what happens, something with volcano and all these things. So we cannot model this. This is a simple model. This is the first time we tried this kind of model. So we just impose artificially these moments of big extinctions by telling the points during the period of extinction. We tell the points that they are not diversifying, they are losing diversity. So we impose a net diversification rate that is negative, and and it gets the values from these 3 fossil curves. These are the most known fossil curves for marine invertebrates in biology. The first one from Sepkowski is not touched. It's just counting all the genera of marine invertebrates found in the paleobiology's database. And then the second curve, the Alroy one, is modified by the sampling effort. So they account for what they call sampling bias, but it's not sure that everything is sampling bias. They say the most recent samples, they have more observations, and so they have more diversity only because of observation. And the third curve is from Sappos, it's the most recent one, and it's not touched, but it's more resolved in time anyway. Because none is perfect, we use 3 different curves to impose these extinctions. And so we run our model with these 3 configurations. So we run our model with these 3 configurations, and for the logistic saturated model and the exponential model. And at the end, we are following the points along the ocean for their lifetime and how they accumulate the diversity. So here I just put the example. Instead of the seafloor age, this is diversity, 300 million years ago and now. So once we run the model, we obtain, the last slide is this, the current diversity in points. And from these points, we can just get the maps of regional diversity just by interpolating into grids of 0.5, 0.5 degrees. So these are our maps. This is the simulation for the first extinction pattern, Sepkoski, but they are quite similar, the outputs. So what we can see on the left is the logistic saturated model. We can see that diversity from top to down is down in time, accumulates in the coastal areas, but it doesn't, the model doesn't allow, doesn't allow diversity to increase in the diversity hotspots that we know today along the tropical area. Instead, the exponential model on the right, it allows diversity to accumulate in the hotspots in the tropical areas. And we get quite nice map with the Indo-Pacific and the Caribbean area hotspots of diversity. Okay, so yeah, I thought the presentation was short, but actually it's a lot of stuff. I'm finishing. So from the spatial maps to pass to get a global curve of diversity, we need to assume some things because we don't know the identity of the genera that are being diversifying in the ocean. So first, we sample each of the 82 maps with transects. So we trace transects from diversity peaks to nearest troughs. And for peaks, we detect them as local diversity maxima in the map that are greater than the 75% of diversity in the map. So you just capture the hotspots of diversity in each map. And then from this peak, I go to the nearest trough, and the nearest trough, so drop of diversity, is diversity in the mid-ocean reaches. That is just one genus. So I get all these transits, more or less 400 by map. And then these transits go from coastal areas with high diversity to the mid-ocean with low diversity. And we integrate diversity along these transits that have to be cut because some of them are very long and some are very short. We just want to do the same sample effort along the map. So we cut them at 500 kilometers. And we integrate the diversity along these transits according to the genera turnover with distance. And this genera turnover with distance, we get it again from bibliography from fossil records. There's a guy, I think it's Miller. Well, there's one paper in which they compare fossil collections, the genera in fossil collections, and according to the distance between these fossil collections. So we get this Jacquard index with distance, so the turnover of species, and we translate it into the proportion of genera in one grid that have to be added to the previous grid in the transect according to the distance between these two. Okay, it's simple, but it's difficult to explain. At the end, we integrate the diversity along this distance, and we obtain diversity along the 400 transects. And the third step is just to integrate all these diversity from the hotspots into the global diversity. And for this, we do the same. We assume a diversity turnover with distance, the same curve. And in this case, we do an integration. I call it like a zigzag integration from the greatest to the lowest diversity. It can be anywhere in the map, according to the shortest distance to the transects already integrated. Anyway, it's a stepwise integration of transect diversity according to distance. At the end, we obtained what we wanted is the global diversity curves. Here I show you from left to right the three extinction patterns imposed and the comparison of our curves with the fossil global curves. So in blue is the exponential model, in red is the logistic saturated model, and in dotted black is the fossil record. And what we can see is what we already knew from the maps, that the exponential model quite fits the fossil record, while the logistic model cannot reproduce this increase at the end of the time series in the last 200 million years. It's like flattened because we don't allow diversity to continue. We put it a limit, okay? But this is theoretically absurd to think that the Earth has no limit. So we just wanted to impose very low values of carrying capacity to the logistic model to test if it was saturated. But what we really want to know is how much the ocean is saturated along its time life of 500 million years. So what we did was to play with the logistic model, changing the limits of the carrying capacity in the regions. We just ran a set of simulations, changing the minimum carrying capacity, so the minimum number of genera that the place in the ocean with less food has, respect to the maximum carrying capacity of the regions with most food. And every time we check the correlation of our output global curve with the fossil curve, without taking into account the extinction periods imposed. Anyway, what we see is that the more we let the model go towards exponential growth, so we let the limits go up, the best the model fits to the fossil observations. And then we do a kind of this thing, we do it for different sets of parameters, and at the end we get all the outputs that fit quite well the data. This is set to a limit of 0.7 of this correlation index. And we set which would be the calibrated logistic model carrying capacity limits from this. We just do this because we want to see the map of how the ocean is saturated, okay? Anyway, we obtain these calibrated curves. They are in between the logistic saturated model and the exponential model, and they fit quite well the fossil curves. Just think that this is super simple assumptions, and it fits, so it's quite nice. I'm finishing, eh? So now I just show you the simulation, so the accumulation of diversity in this calibrated model. It starts 500 million years ago with 1, and then it starts to accumulate. And of course, it accumulates faster in the coastal continental platforms where the water column is shallow and you get the food and higher temperature. And then sometimes you can see that diversity disappears because this is the imposition of the mass extinction events. Anyway, this is just to show you that there's a continuity of the plates are moving, so they are carrying all this diversity. Of course, this diversity is radiating and is adapting to the new environments, but anyway, the pace is changing the pace of this radiation is changing depending on the environment it's crossing this land. Anyway, I think this was just to show off a bit that there was a continuity in the model. To finish, to see how much the ocean is saturated with this calibrated model, we just plot as an index of this saturation the diversity in each grid by the carrying capacity, the effective carrying capacity in that grid, given by the food in the grid, ¿vale? And so we see that most of the ocean is far from saturation, so like near zero, and so it means that diversity can continue because the carrying capacity is much higher. And then only a few spots, the hot spots areas in the ocean, are near saturation. So diversity is close to the limit diversity, the carrying capacity. This is the end, ¿vale? So, sorry, it's very dense. So if we plot these grids, this area, only the coastal areas that are the ones that are more saturated because they carry more diversity. If we plot the histograms of these areas respect to the diversity to carrying capacity fraction, we can see that very few areas of the coastal environment are near saturation. So the ratio of D by K is greater than 0.5. This is kind of, nowadays, is the black dots. It's only like 2% of the global continental coastal ocean that is near saturation. If we put it in the logistic model, these plots would mean that most of the global continental areas are in non-equilibrium. They are growing, accumulating diversity in an exponential way, and only less than 2% of the global flooded continental areas approach the equilibrium, so approach the limit of diversity accumulation. So we kind of answered this long-time question from fossil, or at least we give a tool to answer to this, and we can improve it. But to end, the model also allows to address another long-standing question in evolutionary ecology. That is, it demonstrates that the steep increase in the last 200 million years is real. Vale? And this has been a big debate because it's from fossil records and they have a sample bias. So this increase in the global fossil records that we reproduce with our model, some people said that it was just a cause of sampling bias. The more we approach in time, the better the fossils are preserved, and so we observe more diversity. It's like we go in the ocean and we sample more times, so we get more diversity, okay? Other people say that no, that this was real and maybe related to continental fragmentation. So from Pangea, the continents fragmenting, and this gave to more coastal areas that were that had some barriers, so they gave more diversification, and then they had very different environments. So this is another theory that they were just hypothesizing. And then there was another theory that is the Mesozoic marine revolution. They say that from this big extinction, diversity increased a lot because there was a race of weapons of new forms of life, and this made that the carrying capacity increased because they were occupying the new niches. And then in this paper, we call it the diversity hotspots hypothesis. We say that also this rise is could be just because of the long time that the Earth has been without a big extinction that drops the diversity again, and also the stability of the continent's position has allowed for this development of hotspots in the low latitudes. And this is the end. Sorry, I always speak too much, but So the take-home message is that this is the first globally spatially resolved model of diversification in the Phanerozoic. It can be much improved, of course, like all models. I'm not a modeler, this is my first little model, but I believe in models. And it can be a benchmark to test mechanisms explaining macroevolutionary patterns. There's a lot of hypotheses and always contradicting from fossil records, so we can try to help with this model, improving it. And finally, the diversity hotspots hypothesis. We write it like this in the paper. Diversity has steadily increased in the last 150 million years due to the development of diversity hotspots under prolonged conditions of Earth system stability and maximum continental fragmentation. This is the end. The paper is now in review, so you can read it in Research Square. And the model is in GitHub, so you can play with it. It's in MATLAB, and it's quite simple. It's just the diversification model, and just the output of the other two big models. Well, this is just the model. And this is a big thanks to the team, because this has been a real collaboration with very high-quality experts in their fields. Anyway, we have the UC Riverside, that is the people of the Earth System Model, and then we have the people from Sydney University that have the Paleotectonic Model, and then we have Michael Benton, that is an expert in the theory about all this diversification in the deep time, and then we have Sergio and Guillaume, that are part of the team, and we're helping a lot in setting the diversification model and passing from alpha to gamma. And that's all, and thank you to the funding to Explora. That's all. Thank you.
Thank you very much, Carmen. I think you were perfect in time. So
yeah, because I spoke too much.
No, that was perfect. And we have time for questions now. So if you have any question, I can pass the micro.
Hello, can you hear me well? Well, thank you for the talk. I think I'm not a modeler, and I could follow the talk until the end, so thank you for that.
Thank you.
My question is a bit of coming out of curiosity. From my understanding, your two main parameters into the model are food and temperature. And I can understand temperature, but my question is more parameters or factors. Parameters or factors you're taking into account when you're importing food into the model. Are you taking into account nutrients or population or?
No, it's just, yeah, I spoke too much, but the nutrients is just the particulate organic carbon going to the deep sea. So it's the food available. And the more food, this is a kind of like if you think in population ecology, but maybe you're not from that field. But anyway, the more food you have, the population size is greater. So the possibility of extinction of the species in that place is lower because the population is greater. And the more food, the more the populations reproduce, and the more opportunity for mutation and radiation and so on. So this is the effect of food in the diversification, and the effect of temperature is just the speed of diversification.
A question. You talk a lot about diversification in this paleogeological scale, but you don't talk about which is the target group and how do you measure it and how you sample it,
yeah,
and the extent of the sampling in the different places. And I think that is quite relevant, no, for what you find in this study.
Yes, but as you see, the diversification model is very, very simple. We get parameter ranges for this kind of invertebrates organisms, but at the end, yeah, we are just modeling an entity that is similar to invertebrates in this, that have something to do with the benthonic system. Either they live in the seafloor or they have a stage in their lifetime that they are in the seafloor. But anyway, we are just, we are not focusing in a group. It's just, actually because it's not a group, there's a lot of radiation. This is 500 million years, so it's everything. We focus in invertebrates and we impose all these parameters and the rate of diversification from marine invertebrates, benthonic marine invertebrates, that are the ones that are compiled in the fossil records, in the global curves. But it's a general, this is a very simple model.
But they give a trace in the fossils because they have solid things, like a muscle or whatever, or any invertebrate is in this.
That's why. So the fossil records has a lot of biases because it's super difficult. If we think that it's difficult to sample nowadays our things, plankton, imagine these people of past things. So in general, are organisms that have a skeleton or that they can be preserved. But anyway, this is a compilation of everything, all the marine invertebrates. And the model is very simple in some species that we call it genus here because it's based, the rate of diversification is based on genera fossil curves. But anyway, it's very simplified. Yeah.
Thanks for the talk. I'll pick up from the same question. I have, I'm a microbiologist, and I have a very, very hard time at believing that temperature has anything to do with diversity. And if anything, it will be in the contrary, the way that you are showing. So, and it's the same about nutrients, whatever the nutrients you measure. I mean, I would tend to believe that diversity is related, it's not related linearly with temperature, nor with nutrient availability, at least with microbes. And then I also wonder where, I mean, if I am thinking about microbes, I cannot understand a logistic model. I mean, there has to be an exponential way. And we see it with the COVID every day. I mean, there's new versions and new versions and new versions and new versions.
Yeah.
So diversity in the term of different types of individuals, I mean, microbes, I don't see the limitation. I don't.
That's
so
why the
exponential with that.
Yeah, that's why the exponential model fits better. And also the answer about temperature and food. I'm you. I mean, I'm plankton ecologist and I'm from observation. You have to think that this is deep time. And I really think that temperature has to do, temperature accelerates metabolism and accelerates processes, and so it may accelerate net diversification. It may accelerate diversification, so mutations and everything because of the pace of metabolism and reproduction and everything, and also extinction, and then the net diversification we think is positive, but it's very theoretical. And then the food, in plankton is different. But we are talking about benthos is a limited space area, and it's just an occupation. But as you say, depending on how many niches they occupy, they can fractionate all this. And that's why, in the curve, in the general curve, they explain it a lot with the Mesozoic marine revolution. This is just a revolution that started with Durophagos. I didn't know it, but now I know. It's just a new way of predation that was making holes in the shells of mollusks and things like that. And with this, they started to do this race of arms, and they started to conquer all this sediment in another way, and they created new niches. To say so, yes, I think if there's limit, it may change in time, and that's why in Mesozoic marine revolution, they say that maybe the carrying capacity increased, and so that's why it goes upper than this limit, previous limit. And what we see is that our curve is the blue one, is consistently lower than the fossil record in this time. So I think this revolution, this niche partitioning, making the best of the food or space available made that the diversity increased. So we don't account for this in the model, and maybe that's why we are down here. Or maybe because in the model part of this extinction is already accounted for in the Paleogeographic. But anyway, I agree with you, but...
Yeah, the PT change, what is this? What is that one?
The PT change.
The change between the P and the T, I mean...
You mean these
the T
letters?
goes down and then suddenly it explodes. So what this
Here,
change-
okay, so I think I said, but maybe no. The gray lines are the imposed mass extinctions. This is the Permo-Triassic mass extinction. Almost everything disappeared in the ocean. And new ways of life appeared, and there was a huge radiation.
That's the Burgess shale thing.
So we impose it artificially, and maybe we go from here to here, and the fossil corpse goes from here to here. So part of this go down is already in the model without imposing. So anyway, there are twitches in the model, but yeah, one thing that maybe could be nice, but we wanted to make the simplest model, but of course we can change. We can say, okay, from here we know that there was a revolution in the arms, blah, blah, so we will change the carrying capacity, and these kind of things. You can make complexer and complexer, but you don't want to fit, you want to do simple and try to, yeah. But I agree, it's more exponential than logistic. It's far from saturation, yeah.
Thanks for the talk. But the logistic model assumes that the carrying capacity is constant,
Yes, that's what I was saying.
all the, like throughout the years, constant. But that doesn't have to be like that, right?
Yeah, that's the point. Yeah. We consider a constant carrying capacity that goes between the ranges that we impose. These ranges are from the food, and the lower range is the minimum in the map of food, and the other is the maximum in the map. So it kind of resets for each map, but we are not changing the carrying capacity because of ecological innovation or no. It's fixed. Because there's been a big debate on, I don't think it's fixed, but this is the simplification. But the people that are saying it's saturated and all this, they in their mind is a saturated logistic model. It's fixed, and diversity is near its carrying capacity, and this is not true from the model. So, yeah.
Thanks.
Thank you very much for the talk, Carmen. Concerning the model, I know that you said that the simpler the better, but is there anything that you will include to improve this model?
More than improving, answering to the, now we are answering to the revisions. So when, for example, to say that this model is quite nice is that a reviewer said, why not doing simpler? Just fix and just account for the time. So today the seafloor is for 200 million years here and there. Why not fixing it? And the output curves were, well, I can show you, but anyway, they were very simplified and nothing to do with the fossil record, okay? And then answering to another reviewer, they were talking about this Mesozoic marine revolution, and they were saying how you add your new hypothesis to this revolution. And I thought of this inconsistency of the fossil record and the model, and I thought maybe in the future we can, as I said to Pep, just give more than greater carrying capacity, greater diversification rate from this point, because we know that they explored new ways of life and the radiation was faster. So you can improve, but I'm not sure, because already to do this model, we do big assumptions. So everything that you want to improve is making another big assumption. And I'm not sure. But just turning on and off this thing of changing the net diversification rate with time could tell us if the marine revolution improves the model, or I don't know. But anyway, I'm not a modeler, so.
Good for you.
Thank you. I like it a lot, and I also followed it. I think I may have missed this from the last part. Do you arrive until the present? So can you reproduce the present living benthic diversity?
Yeah, it's the 0 time. So actually, it's the 0. Well, the current is the current position of the. Is the last time. No sé dónde está aquí. So the last line is 0 million years ago.
So
Is now.
This is not
This is now.
fossil.
And yesterday, so another revision answer is, there's one reviewer that says we need to compare with fossil records spatio-temporally and blah, blah. And we try to argue that this is not very helpful because fossil records are very patchy and it will be very difficult testing the model. Even if it fits or not spatially, it doesn't say much. But we said, okay, we contacted a girl that works on global diversity of benthic invertebrates. We asked her to give us data from OBIS. But we were trying, I will not show you, because yesterday we were trying. The problem is that she was trying all crustaceans. And of course, she showed me the map, and it had a lot of diversity in the middle of the ocean. And I told her, well, but most of these are plankton, and most must be copepods or whatever. Yeah. So it's not much point because we are modeling the deep, the benthic. So we were trying to check only the coastal areas, but still she has problems with sampling bias. So we are working on that. But just from publications, previous publications from other people, this was maybe what we should do is just to scan these plots and test, because actually it's quite similar. In big things. We get the high diversity here and a bit lower in here. And then in the Mediterranean Sea, we get a big spot, a hot spot, but maybe one part is due to the model limitations, the sea Gini limitations in space. It's big, very big grids, so it doesn't. But anyway, in the paper we will put a comparison with the
actual.
Yes.
Thank you.
Last question, perhaps, or perhaps not. Okay, so I think we can leave it here. Thank you very much, Carmen. It was very, very interesting.
Thank you. Sorry, I speak a lot. I get stressed.
You didn't speak a lot. You did just right.
Okay.
So thank you very much, and everybody.